Golden Helix is pleased to announce that the Oncology Mutation Database, or OncoMd, is now integrated with our VarSeq software. In this video, we will describe VarSeq and OncoMd and give a brief demonstration of how they may be used together. The VarSeq software from Golden Helix is a powerful platform for analysis of DNA sequence variants from many types of sequencing experiments, including the analysis of somatic mutations in cancer. Among its many features, VarSeq provides an extensive library of carefully maintained public annotation sources to provide doctors and scientists with the information necessary to interpret variants. The Oncology Mutation Database, or OncoMd, is a repository of cancer genomics data curated by MedGenome. OncoMd is frequently updated and currently contains information about more than 2 million variants gathered from over 7,000 peer-reviewed publications. OncoMd includes valuable data about the clinical interpretation of somatic mutations, including information about drugs that target specific mutations and treatment response rates for those drugs. It also keeps a catalog of ongoing clinical trials for cancer treatments that target mutations in specific genes. The incorporation of OncoMd annotations in VarSeq gives oncologists and cancer researchers a powerful new tool for sequence interpretation. In clinical settings, OncoMd can be used to identify targeted therapies that may benefit a certain patient or to identify clinical trials for which the patient may be eligible. For research purposes, OncoMd provides scientists with links to research papers about specific genes and variants, together with rapid access to bioinformatics resources from NCBI and elsewhere that may assist in the interpretation of research results. Now, let's go ahead and take a look at VarSeq and OncoMd. Here we see a VarSeq project containing data for a tumor sample that was sequenced using an ion torrent comprehensive cancer panel. VarSeq starts from VCF files, and in this case we see that there are nearly 5,000 variants captured in this particular patient's VCF file. VarSeq gives users the ability to apply annotations from a variety of different sources, and then to create logical filters to use that annotation data to subset those variants to a set of mutations that are expected to influence the disease of interest. In this case, we've already created a set of filters that reduces the data to just 18 potential somatic mutations for further review. VarSeq gives a number of useful annotations here that help us get started on this process. We can see what gene each variant comes from. We also see information about the transcript, what is the coding change and the amino acid change that results, and so on. From this point in clinical interpretation, the OncoMd database becomes extremely valuable in helping us to learn more about the context of each of these mutations. As we scroll through the table now, we'll find a number of those OncoMd annotations. First, we see the gene information set. In this column, we can learn a little bit about the individual genes and see some of them are known to be oncogenes, others are known to be tumor suppressor genes. There are also external hyperlinks that will take us out to additional information from sources like Hugo or OMIM. The next table shows us information about the individual variants themselves and what tissues they've previously been observed in. For example, we see some of these variants have been observed in colorectal cancer, prostate cancer, and others. Now, the next set of annotations is especially valuable in the clinic. In this area, we see information about which of these variants may be targeted by particular drug therapies. Only one of our 18 variants has a known drug associated with it, and we can learn more by just clicking on the row of the table, and we'll get a more tabular view down here at the bottom that we can look at. So here we see that with this particular mutation, this happens to be BRAF V600E, which is a very pervasive mutation in many different types of cancer. We see a number of different therapies that are available and also some information about the response rate. how many samples have been evaluated for each treatment, and how many of them responded or did not respond. Now at this point, as a clinician, I may want to start taking some notes. And VarSeq provides a very nice interface for taking notes along the way. And to start with, I'd like to capture some information about this particular variant. So I'll just copy these first few sets of annotations so I can paste it in up here, and now I know what mutation it is that we're looking at. Now further down, I'd like to capture this information about the available treatment options. Select that, paste it in. And now I have that information all ready to go. I can save it out as a PDF file or an HTML file so that I can pass it along to the referring physician. Now, what else can we learn about these mutations? Well, as we move through the table, we see that the next set of annotations tells us about clinical trials. This particular patient has mutations in ALK and in RET, both of which have open clinical trials available that the patient may be qualified for. If I select the ALK mutation, for example, we see here at the bottom that there are clinical trials currently recruiting patients in both the United States and Canada for particular types of brain cancers, lymphomas, neuroblastomas, and so on. There is an external link here that I can use to get to more detailed information about each of those trials. Finally, if I'm a researcher learning more about the individual variants and about the profile of this individual tumor, this last section will give some very helpful information where I can see, for example, that there are publications describing this particular mutation in PDE4-DIP and some of the other genes here as well. There are links to PubMed and other external sources where I can learn more about those variants. So with that, I'd like to thank you all for joining us today. If you have any more information about VarSeq, about OncoMd, or about Golden Helix, feel free to contact us through our website at goldenhelix.com or email at info@goldenhelix.com. Thank you.